In 2023, Harvard University, the Smithsonian Institution, and 23andMe did something that had never been done before. They sequenced the full genomes of 27 enslaved Africans buried in a forgotten Maryland cemetery and matched their DNA against 9.3 million living Americans. They found 41,799 genetic relatives. That number is just the beginning. When scientists began systematically comparing the DNA of enslaved Africans against every modern population on Earth, from burial grounds in South Carolina to rice plantations in the Low Country to ironworks in the mountains, the results contradicted what researchers expected at nearly every turn. Hidden trade routes, documented crimes written in chromosomes, families buried side by side that nobody knew existed, and a genetic record of the slave trade that is more accurate, more specific, and more devastating than anything the ship manifest ever captured. Before we dive in, comment where you are watching from and click subscribe. To understand why this research is so extraordinary, you need to first understand the problem it is trying to solve. Between 1619 and 1865, somewhere between 400,000 and 500,000 Africans were brought directly to what is now the United States as enslaved people. Globally, the transatlantic slave trade forcibly moved more than 12.5 million Africans across the Atlantic between 1515 and the mid-19th century. The record-keeping that existed, ship manifests, port logs, plantation inventories, tracked cargo, not people. It recorded the port where a ship departed in Africa. It almost never recorded where the individual people aboard that ship were actually born, what ethnic group they came from, what language they spoke, or what part of the continent they had been torn from. When those people arrived in the Americas, most had their names erased or changed. Their ethnic identities were deliberately obscured. Their languages were suppressed. Families were separated, sold apart, scattered across hundreds of miles with no mechanism to ever find each other again. And the records that might have preserved who they were, who they truly were, not what their enslaver called them, were either never created or destroyed or locked away in archives that nobody thought to search. The result, for tens of millions of descendants of enslaved people living in America today, is a wall, a specific, deeply personal place in their family history where the names stop, where the records end, and where the question of where their ancestors came from goes completely unanswered. Unlike the descendants of European immigrants, who can often trace family lines to specific villages in Ireland, Poland, or Italy, who can look up a great-great-grandmother in a church register and find her parents' names, her birthplace, her occupation. Many African Americans hit that wall somewhere in the 1700s or the 1800s and find silence on the other side. Not because the people were not real, not because they did not have names and homelands and histories, but because a system was specifically designed to strip those things away and make sure they could never be recovered. The question driving this entire field of science is, can DNA see through that wall? And the answer, across study after study, burial ground after burial ground, is yes. But what it sees on the other side has repeatedly stunned the researchers doing the looking. The first unexpected result came not from the bones of the enslaved, but from the living. In 2015, a landmark study published in the American Journal of Human Genetics, led by population geneticist Catoctin Furnace, in collaboration with David Reich's lab at Harvard Medical School, using data from 23andMe, analyzed the genetic ancestry of more than 160,000 Americans. The findings on African American ancestry were precise. On average, the African American genome is 73.2% African, 24% European, and 0.8% Native American. The 24% European figure is the one that stopped researchers cold. Not because it was unknown in a general sense, but because of what the genetic analysis revealed about how it got there. The European DNA flowing into African American genomes was not evenly distributed between male and female ancestors. It was heavily, consistently, measurably skewed toward European male ancestors. The signal was strongest on the Y chromosome, the chromosome passed exclusively from fathers to sons. Researchers at McGill University, building on this finding, were able to use genetic modeling to pinpoint approximately when much of this European ancestry entered African American family trees. Their conclusion: the overwhelming majority of the European ancestry in African American genomes entered before the Civil War, during slavery, during an era when black women in the United States had no legal protection whatsoever from assault by white men, including the men who legally owned them. The DNA had encoded the crime across generations of living people in every state in the country. The sexual exploitation of enslaved African American women, which historians had documented, which survivors had described, which descendants had always known, was now also written in the genome, permanent, molecular, undeniable. But the 2015 study only looked at the DNA of the living, people whose ancestry had been mixed and blended across centuries of generations. To get closer to the source, to look at DNA that came directly from people who had recently been brought from Africa before mixing, had blurred the signal. Scientists needed ancient DNA, the DNA of the enslaved themselves. And that's where the next unexpected result waited. In August 2023, the journal Science, one of the most prestigious scientific publications in the world, published a study that represented a genuine first in human history. A team led by Edwin Harney of 23andMe, with David Reich of Harvard and the Smithsonian Institution biological anthropologists Kari Bruoheide and Douglas Owsley, sequenced the full genomes of 27 enslaved and free African Americans buried at a place called Catoctin Furnace in western Maryland. Catoctin was an iron forge that operated from 1774 to 1850. It produced weapons for the Continental Army during the American Revolution. It was powered throughout most of its history by the forced labor of enslaved people whose names were tracked in ledgers the way equipment was tracked, as property, not as human beings. When a cemetery tied to the furnace was excavated during highway construction in the 1970s, the remains were transferred to the Smithsonian. For decades, they waited there, unknown, unnamed. The study published in 2023 changed that. The ancient DNA extracted from the Catoctin bones was compared against the genetic profiles of 9.3 million consenting research participants in 23andMe's database, people living across the United States, across sub-Saharan Africa, and across Europe. The results rewrote what researchers thought they knew about these specific individuals and about early American slavery more broadly. The first unexpected finding was about African origins. Historical records about the early American slave trade pointed heavily toward West Africa, particularly the coastal regions of Senegambia, Sierra Leone, and the Gold Coast. These were the ports that appeared most frequently in the shipping manifests of the 17th and 18th centuries. So when the Catoctin genomes were compared against African populations worldwide, researchers expected the matches to cluster around those coastal West African regions. They did, but not only there, and the specific matches were far more precise than anyone anticipated. The strongest African genetic connections for the Catoctin individuals were to two very specific ethno-linguistic groups, the Wolof and Mandinka of Senegal and the Gambia in West Africa, and the Congo of Angola and the Democratic Republic of the Congo in Central Africa. These two homelands are separated by more than 3,000 miles of African coastline, yet both were represented in the same small rural Maryland cemetery. The Wolof are one of the most culturally distinct peoples of West Africa, a people with a rich tradition of oral history, Islamic scholarship, and a complex social hierarchy whose griots preserve genealogies and community memory across centuries. The Congo were a powerful, historically literate civilization in Central Africa, a kingdom that had diplomatic relations with Portugal as early as the 1480s, that produced its own written records and whose people were targeted in enormous numbers, particularly in the final decades of the transatlantic trade when Central Africa became the single largest source of enslaved people transported to the Americas. Together, the Wolof and Congo people represent two of the most historically significant African cultures drawn into the slave trade. Seeing both groups represented in the same small rural Maryland cemetery is a testament to the geographic reach of a commerce that assembled human beings from opposite ends of a continent and forced them to forge iron together in the Appalachian foothills. If you are as gripped by this research as I am, the science that is literally giving names back to people history erased, subscribe to this channel right now. This is what we do here: facts, science, and the stories that got buried alongside the people who lived them. Hit subscribe. The second unexpected finding was about European ancestry, and it was the most disturbing result in the entire study. Most of the 27 Catoctin individuals had some European ancestry in their genomes. That was not surprising on its own. Historians knew that mixing between African and European populations had occurred throughout the slavery era. What was unexpected was where in the genome that European ancestry was concentrated. It was strongest on the Y chromosome. The Y chromosome is passed from fathers to sons, and only from fathers to sons. A strong paternal European signal means that the European ancestors of these individuals were overwhelmingly men. Their DNA entered these family trees not through marriages or mutual relationships, but through the sexual assault of enslaved black women by white men. Harvard Magazine, reporting on the study, stated this plainly: the European signal in the Catoctin genomes was the most direct genomic evidence yet of the sexual exploitation of enslaved African American women by their European American enslavers. This wasn't a statistical inference. It was written directly into the structure of the DNA, into the chromosomes of people who had been enslaved in Maryland in the 1780s. And it matched almost precisely what a separate computational study from USC and Stanford University found in 2023 and published in the journal Genetics. That study showed that a randomly selected African American born between 1960 and 1965 descends from, on average, 314 African ancestors and 51 European ancestors, and that the European ancestors appear in family trees overwhelmingly during the era of slavery, not after it. 314 African ancestors, 51 European ones, a number that tells you the scale, a ratio that tells you the mechanism. The third unexpected finding from the Catoctin study came when researchers cross-referenced the 27 ancient genomes against the 9.3 million living people in the 23andMe database. They found 41,799 genetic relatives. Let that number sit for a moment. Not 41,799 vague ancestral connections from thousands of years ago. Not distant cousins separated by dozens of generations and multiple continents. These are people alive right now in the United States who share specific, traceable segments of DNA with individuals who were enslaved in Maryland before the United States was even a country. People who are probably going about ordinary lives, driving to work, raising children, watching the news with no idea that buried somewhere in their 23andMe results is a thread connecting them to a man or woman who forged iron in the shadow of the Blue Ridge Mountains and died without a name on their grave. Among those 41,799 relatives, 2,975 shared long enough stretches of DNA to be considered meaningfully close relatives, rather than simply distant ones. And the highest concentration of those close relatives, still in Maryland, more than 200 years after Catoctin Furnace enslaved their ancestors, some descendants never left. 3 individuals buried at Catoctin also carried gene variants associated with sickle cell disease. This genetic condition was shaped by thousands of years of African history, by the malaria-driven natural selection that favored the sickle cell trait in West and Central African populations. It was carried by these 18th century Marylanders, and it is carried by living African Americans today. It is a biological thread 250 years long, running from an unmarked grave to a modern doctor's office. The fourth unexpected result came not from ancient DNA, but from the living. and it came from the 23andMe mega study published in 2020 in the American Journal of Human Genetics, the largest genetic investigation of the transatlantic slave trade ever conducted, involving 50,000 people with African ancestry across the Americas. The study's core goal was straightforward. Compare the genetic ancestry of living people of African descent in the Americas against the historical shipping records of the slave trade and see where they agreed and where they disagreed. The disagreements were the story. Population geneticist Stephen Micheletti, who led the study at 23andMe, described their astonishment. While much of the genetic data agreed with the documented history, in some cases, the DNA and the ship manifests diverged, and the divergence was striking. Nigerian ancestry, specifically connections to the Yoruba, Isan, and Igbo peoples of Nigeria, was dramatically overrepresented in the United States and Latin America compared to what the shipping records would predict. The manifest showed relatively few ships coming directly from Nigerian ports to American shores, but the DNA said otherwise. Researchers worked with historians to find the explanation, and it turned out the manifests were telling only half the story. Enslaved Nigerians were frequently shipped first to the British Caribbean, then traded again into the American South through an internal slave trade within the Americas that the transatlantic records never captured. The DNA saw through the gap in the historical record. It found the hidden trade route. It found the people the manifests had erased twice. First by listing their departure port incorrectly, and second by not recording their final destination at all. Meanwhile, ancestry connected to Senegal and the Gambia, the Senegambia region, showed up far less in the genetic record than the shipping logs predicted. This had an explanation, and it was devastating. Enslaved people from Senegambia were disproportionately sent to rice plantations in South Carolina and Georgia because they had expertise cultivating rice in their home regions. Enslavers wanted to exploit that knowledge. Those rice plantations were among the most disease-ridden environments in colonial America, rife with malaria and other tropical illnesses that killed at catastrophic rates. The enslaved people who survived the Middle Passage, who endured the indignity and violence of the auction block, who were marched to the Carolina Lowcountry, many of them died there within years of arriving before they could have children who would carry their DNA forward. The death rates were so extreme and the demographic destruction so thorough that their genetic contribution to the modern African American population is measurably smaller than the number of ships that carried them would predict. Their underrepresentation in today's genetic record is the molecular signature of a mortality rate so brutal it nearly erased an entire West African people from the genetic future of the Americas. The DNA did not just tell where enslaved people came from, it told who survived and why. Taken together, what do these studies, the Catoctin Furnace Project, 23andMe mega study, the 2015 Ancestry analysis, and USC and Stanford genealogical modeling actually reveal that nobody expected? They reveal that the shipping manifests of the slave trade were systematically incomplete, and that DNA is a more accurate historical record than the documents the slavers left behind. The genetic evidence exposes hidden trade routes, hidden populations, and hidden crimes that the paper record either never captured or actively concealed. They reveal that the sexual exploitation of enslaved black women was not an incidental feature of American slavery. It was so pervasive, so systematic, so multi-generational that it left a measurable, directional, sex-biased signal in the genomes of tens of millions of living people, a signal strong enough that researchers can detect it in the Y chromosomes of 18th century bones. They reveal that the people erased by slavery are not truly gone. They are genetically present in the living relatives who share their DNA, in the ancient samples that preserve their ancestry, in the sickle cell variants and disease risk factors, and ethnic group connections that stretch across centuries without interruption. And perhaps most powerfully, they reveal that studying the DNA of enslaved Africans and comparing it against every modern population on Earth does not produce tidy, expected results that confirm what the history books said. it produces surprises. It finds the hidden connections, the suppressed routes, the populations that the records undercounted, and the ones that mortality nearly silenced. It finds the families buried side by side. It finds the 42,000 living cousins nobody knew existed. The science is not finished. The databases of African genetic profiles still have enormous gaps, entire ethnic groups and regions whose genetic signatures have never been cataloged. The number of ancient genomes from enslaved individuals remains vanishingly small relative to the 12.5 million people the trade moved across the ocean. The ethical frameworks for this work, ensuring that descendant communities drive the research rather than being studied as objects, are still evolving. But what is already clear is this: the slave trade tried to make these people nameless, placeless, genealogically severed from everything that had made them who they were. It tried to reduce them to property in a ledger. It almost succeeded. The DNA refused. It has been refusing for 400 years, sealed inside teeth and bones and the living cells of every descendant, quietly carrying the record of names and homelands and family bonds that the slave trade spent centuries trying to permanently erase, and waiting for science to finally develop the tools to read it. Please subscribe if you want more of these videos.